Hey, welcome, hello there, to one of our anatomy classrooms where we have models. This is not the cadaver lab. We do have a cadaver lab with 3 cadavers, but we first start out with the skeleton and skeletal models. And so when you first learn anatomy, you learn a lot of terms like lateral and medial and superior and inferior and transverse and coronal and sagittal. There's a lot of terminology that we cover in anatomy. Then you learn about bones and their specific landmarks, how muscles attach to the bones. And you can see the red and blue lines on this skeleton are showing us where different muscles originate or insert. Here we can see, how along the spinal cord that there are spinal nerves, that the way the vertebrae stack together, there's a little space between each vertebrae called an intervertebral foramen, and you have spinal nerves that come out there. So if one of your discs bulges, like this one here, it can put pressure on a spinal nerve and cause a lot of back pain. And sometimes that requires operation, sometimes not. We have Mr. Muscle Man, where you can study the muscles on a model, like pectoralis major, rectus abdominis, or external oblique, or the deltoid, the biceps, and so forth. But we also look at these muscles on a cadaver. So, and it looks quite different when you get to the cadaver. So we use models, we use 3D anatomy software, we use our textbook, we use the cadavers quite a bit, and we try to go system by system. So we do skeletal system, muscular system, integumentary system, digestive, reproductive, urinary, respiratory, and so forth. So that's kind of the general approach that we take, and we just keep building and building and building, essentially trying to make a 3-dimensional map of the body, so that if you think about any point on the body, you can think about the different systems, the different layers, the blood supply, the nervous supply, the muscles, and everything that's in that region. That's the basic approach.
Okay.
Some other basic supplies, everybody seems to like pipettes. This can accurately measure between 5 and 50 microliters. So you can just turn the little dial, and if I want 26.5 microliters, I set it to that. I get my little pipette tip and dip it in and let go. And 26.5 microliters is just a teeny tiny drop. So this allows you to basically conserve supplies by, it's like taking your cookie recipe and dividing it by 1,000. And you just need teeny tiny components. Of course, you'd end up with a very small cookie, but that's for measuring those very small quantities. And what else do we have? Some electrolysis for doing understanding proteins and the different parts of proteins. That's called gel electrophoresis. If you're wanting to separate a protein into different components using an electrical charge. You have a lot of microscopes here. Various reasons to look under a microscope. A lot, if you're doing microbiology and you're trying to figure out what a certain microbe is, you're going to have to look under the microscope, describe the shape of the cells, maybe do different staining techniques to see, is this gram positive, is it gram negative? And it's difficult with bacteria. Bacteria tend to all look very similar to each other. So step by step by step doing diagnostic techniques to try to figure out what you actually have. And even then it's quite tricky. Nowadays we can cheat and send bacteria off to have their DNA analyzed, and you can cheat and discover what species you have that way.
Different than the one you would have at home.
But we use fume hoods the scales. This lab is used for microbiology and general biology and physiology. So if we're working with chemicals where we need to be careful with the gases, then we always work under a fume hood. We have hot plate and stir plate. It's kind of fun. You can put your solution to heat up, and you put this little thing in there, and when you turn it on, it will actually stir. You can get it to, it'll stir your solution for you. So imagine a bottle of, or a glass of your solution that you're mixing together while you heat it. That will just stir away. As we do general biology and we look at the kingdoms of plants and fungi, and study different types of fungus. A lot of what we do is for microbiology. For example, these little heaters. This you plug this in, it gets really hot. If you use this, you have little metal loops in microbiology. So you put that in there for just a little bit and it sterilizes it, and then you can put your sample onto a growth plate, onto the auger, and you know that since you've been in here, you've sterilized, so then when you dip into your sample, you're only getting that sample, not any contamination. And then you sterilize again. You keep sterilizing. All right, so we have a variety of little creatures here that like to live in the soil and help decompose things. Large millipedes, hissing cockroaches. Pretty much they just do their own thing. You know, you wouldn't, they're not going to hurt you in any way. Some people find them a little too exotic for their tastes, but it's just not something we're used to encountering perhaps, but totally harmless. They don't even stink. They don't bite. You also have a lot of these like pill bug things, potato bugs people call them, roly-polies. So in our general biology lab, you can do little experiments with them, learn about their classification, their lifestyles. Did you know that praying mantises are actually related to cockroaches fairly closely? And praying mantises get a much better reputation. But cockroaches, they're as ancient as they look. They've been around a very long time. It's a design that works remarkably well. They're a type of beetle, and this particular species doesn't have wings, which is unusual for an insect. But otherwise they're a fairly typical insect with their 6 legs, their antennae, their different segments, and head, thorax, abdomen, and so forth. Whereas a millipede is not an insect. This is it's an arthropod, but it's an unrelated arthropod. And millipede means 1,000 feet, which I haven't counted, but be good for, if a millipede walks into your shoe store, it's a good day.
Down there, that.
Thank goodness.
Welcome to the Cadaver Lab, where mortu vivos docent, the dead teach the living. So basically, we work on cadavers here. We have 3 cadavers. These are people who've donated their bodies that they lived and died along the Wasatch Front. They donated their body to University of Utah Medical Center, and one of the uses is for students to learn anatomy. This is a great gift to us because each body is unique, and so we can compare the anatomy between bodies. And this is where the students really learn the most, and they really have strong memories from working on the cadavers and understanding how things are positioned relative to each other and so forth. So we're really grateful for the people who make their bodies available. Quick story, I had a cadaver one time. We opened up the bag. We have to dissect the cadavers first. And so we opened up to get started, and there was a note tied to his toe. And I undid the note, opened it up, and said, "Hi students, my name is Boyd, and I was an aeronautical engineer, and I learned that the best way to learn about something was by taking it apart. So I'm all done with my body. Have a lot of fun, learn as much as you can."
Oh, that's cool.
And so he left that little note for us, and so we do. We try to learn as much as we can over the course of the year, and we do take apart the bodies and see how the different parts are put together. And then at the end of the year, the bodies are returned to the University of Utah. They get cremated. Ashes get returned to the family if the family wants them, or there's a common grave at the University of Utah. Pretty much we go down and we are looking, an ideal cadaver for us is kind of medium weight, because let's face it, when you're dead, as you're dying, you're not usually in good health. And so some people, they've spent a lot of time bedridden, wasting away, and their muscles are really small. So we look for people who still have some weight to them, because then they're still going to have muscles. That's kind of our first criteria. For female cadavers, it's nice if they still have a uterus intact. So we look to see what scars, and it's fine if they've had a hysterectomy, but we try to find at least one cadaver that has a uterus. It's always a guess, as to whether or not a person's going to have their appendix or their gallbladder. That's just some of the variation that we see. Other than that, yeah, it's just kind of the practical question of, is this cadaver going to be easy enough for us to transport? And other than that, yeah, we're not too picky. We either get 2 males and a female, or 2 females and a male. And yeah, so we have different ages of cadavers. And this year we have a younger cadaver who died at 50, and our oldest cadaver is 84, and she actually looks like she was in about as good of shape as the 50-year-old. So anyhow, yeah, we come here each week for lab after we've done the skeletal system, after we've worked on the models a bit, and we just work through one body system after another. The preservation process, the embalming process, hardens all the body parts quite a bit. So, for example, we have brain specimens, and a preserved brain is quite firm, and you touch it and it seems like, oh yeah, my brain must be pretty strong. But a fresh brain is more like holding jello, and your skull is good. It protects that custard, but too much of an impact and you can see how brains just would splat. So take care of your head.
At first it was really fun to take home stories and tell my kids that I had bought cow's eyes, bovine blood, dogfish sharks, what else? Just some pretty weird and interesting things now. And you can see off to the side I have, not all of them yet because we're still working on it, but mostly what each thing is used for which lab, written off to the side, so that might help you.
That would help.
Okay. Go ahead.
Okay, I buy the hissing Madagascar cockroaches. I haven't tried it yet, but if you pick them up, they will hiss, otherwise they don't. Grant says I can, but I haven't dared do it yet. And the millipedes, and I buy best bugs for biology 1610, the lab is 1615. I also buy some dogfish sharks that are pregnant, and Skittles. If you want to know what the Skittles are for, you'll have to take the class.